we take into account whether if these sublethal concentrations can also cause damaging effects on the other species of nematodes and on the ecosystem processes such as decomposition. So my study is the effects of sublethal copper concentration on to the fitness of Lactus acuminatus and the decomposition of Euroclua indica. So this is the nematode use, and the paragraphs or the Euroclua indica obtained from the near the MS-40 house. Nice. So this is the interactions between soil communities. So the nematode use is a bacterivorous CP2 group which feeds on microorganisms, and these microorganisms are the primary decomposers of the ecosystem, which mainly facilitate processes like decomposition and nutrient cycling. So at the presence of inclusion, it can directly affect the nematode population itself, the microbes, and the interactions between the soil ecosystem. So the objective now of my study is to determine the species fitness as indicated by the abundance of Lactus acuminatus at different sublethal copper concentrations. And then my second objective is to determine the decomposition rates of leaves, Euroclua indica at very sublethal sea levels. So in relevance to other studies, we have like the previous article that I have mentioned a while ago. I hypothesized that the increase in sublethal copper concentration would result in an increasing trend in the nematode population because of the known sensitivity of the genus factors. And then for the second hypothesis, the decrease in abundance will positively affect microbial populations due to decreased predation and thus leads to higher decomposition rate. So this is the methodology. So first, the picture for Lactus acuminatus is shown, and then we obtain preparations for we selected leaf species samples here in Iligan, and then we determined the fastest leaf with the fastest decomposition rate, and so we have chosen paragraphs or Euroclua indica. Then for the next. So the inoculum is the bacterial fungal inoculum is the food for the nematodes. So we test it in we obtain culture of the bacteria or fungus which is in the leaf, and then we soak it in 10 grams leaf with 90 ml distilled water, and then we open the inoculum on the broth, and then serial evolutions